Now in this video we discuss about the next phase of the glycolysis, or you can say the productive phase of the glycolysis, that is payoff phase. In the payoff phase, 4 ATP is formed and 2 NADH is also formed from these steps of the glycolysis. And so it is very important step of the glycolysis. The most of the reactions are reversibles except the last step of the glycolysis. So through the payoff phase, the pyruvate is formed. After pyruvate, formation of the pyruvate transform into acetyl CoA by oxidative decarboxylation process. So in this video we discuss on the focus on the payoff phase of the glycolysis. So the first step of the payoff phase of the glycolysis. So as we know, the glyceraldehyde 3-phosphate is formed in the preparatory phase of the glycolysis. Now this glyceraldehyde 3-phosphate enter into the payoff phase of the glycolysis. So this is the structure of glyceraldehyde 3-phosphate. So this is the 3 carbon, that is why it is called 3-phosphate. So glyceraldehyde 3-phosphate is transformed into 1,3-bisphosphoglycerate in presence of NAD+. So NADH is formed from 1 glyceraldehyde 3-phosphate. So we know, one molecule of glucose generate 2 glyceraldehyde 3-phosphate. So that is why 2 NADH is formed from 2 glyceraldehyde 3-phosphate. So this reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate is very important. The mechanism of the glyceraldehyde 3-phosphate dehydrogenase reaction we discuss later videos. So in this video we focus on the steps of the glycolysis and what are the product of the glycolysis and what are the enzymes. So we have to remember the name of the enzymes, the product of the each step, and also the substrate, and which step generate ATP, which step consume ATP, which step generate NADH is very important. So the value of the del G, in the case of the payoff phase, the value of the del G is 6.3 kg per mole. It is a type of endergonic or exergonic. It is an endergonic reaction because the value of the del G is in positive value. Now ATP formation. So the next step is very important because in the next step is a first substrate level phosphorylation step. During this substrate level phosphorylation, 1 ATP is formed from 1 glyceraldehyde 3-phosphate. So this is the structure of 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate with the help of an enzyme phosphoglycerate kinase. Phosphoglycerate kinase transform into 3-phosphoglycerate. So this is the 3-phosphoglycerate. So 1,3-bisphosphoglycerate is transformed into 3-phosphoglycerate with the help of an enzyme phosphoglycerate kinase. Mg2+ is needed for the function of the phosphoglycerate kinase. The most important thing is the formation of ATP. So the seventh step in the seventh step of the glycolysis and second phase of the step of the payoff phase generate ATP. And this 6th and 7th step of the glycolysis is energy coupling reaction. So ATP is formed from 1 glyceraldehyde 3-phosphate. So 2 glyceraldehyde 3-phosphate generate 2 ATP in this step. The value of the del G 18.5 kg per mole. Now the third step or 8th step of the glycolysis is 3-phosphoglycerate transformed into 2-phosphoglycerate in presence of an enzyme phosphoglycerate mutase. This enzyme also needs Mg2+ for its function. So 3-phospho in the 8th step of the glycolysis and third step of the payoff phase, 3-phosphoglycerate is transformed into 2-phosphoglycerate in presence of an enzyme phosphoglycerate mutase. Now the next step, that is 9th step of the glycolysis and the 4th step of the payoff phase. This is the reversible step. This is also the reversible step. So as you can see, all the steps of the payoff phase is reversible except the last one, that is transformation of phosphoenol pyruvate to pyruvate. We discuss later. So now 2-phosphoglycerate to phosphoenol, 2-phosphoglycerate to phosphoenol pyruvate. So 2-phosphoglycerate is transformed into phosphoenol pyruvate. This is a dehydration reaction. So particularly this phase, in this step of the glycolysis, H2 is released out. So 2-phosphoglycerate is transformed into phosphoenol pyruvate in presence of an enzyme enolase. This reaction releases H2O. So this is the dehydration step of the glycolysis. The value of delta is 7 kg per mole. So now the next step of the glycolysis, that is second substrate level phosphorylation. Now there is an important thing. What is the difference between substrate level phosphorylation and respiration based phosphorylation? In case of respiration level linked, substrate level, what is the difference between substrate level phosphorylation and respiration linked phosphorylation? In case of substrate level phosphorylation, the enzymes are soluble enzymes are involved in the case of substrate level phosphorylation. But on the other hand, in case of respiration linked phosphorylation, like ETS cycle oxidative phosphorylation, so in those cases the enzymes are membrane bound enzymes are involved, like complex 1, 2, 3 we discussed in the ETS cycle, ETS electron transport chain topic. So in the case of respiration linked phosphorylation, the enzymes are membrane bound. On the other hand, in case of substrate level phosphorylations, the enzymes are soluble enzymes. So the 10th step of the glycolysis is a second substrate level phosphorylation. So this is phosphoenol pyruvate is transformed into pyruvate, and this reaction is catalyzed by pyruvate kinase. Pyruvate Mg2 plus or Mn2 plus and K plus is needed for the function of the pyruvate kinase. So pyruvate kinase needs Mg2 plus, Mn2 plus or K plus for its function. So this is the pyruvate. This is the enol form of the pyruvate. So when the phosphoenol pyruvate generates, when the pyruvate is formed from phosphoenol pyruvate, it remains in its enol form, but later tautomerized to the keto form. It is enol form and keto form of the pyruvate, and it is a non-enzymatical reaction. So it is the enol form, later it transforms into the keto form of the pyruvate. So there is no enzymes involved. This is a tautomerization process. So the 10th step and the 7th step, in which 4 ATP is formed from one glucose molecule and 2 NADH is formed from the 6th step of the glycolysis. So the net 2 ATP is formed and 2 NADH is formed from the process of the glycolysis. So the calculations of the whole glycolysis we discuss later in the videos. Thank you.